Hey everyone, welcome back to Blackboard Biology. In our previous video, we went over cellular respiration from the aerobic standpoint, using oxygen. But in this video, we're going to go through anaerobic pathways. Now, technically speaking, there's a few different ways that this can occur, and there's a true anaerobic cellular respiration where instead of giving the electrons at the very end to oxygen and making water, there are bacteria that can actually give those electrons to other molecules. But that's not actually what we're going to look at in this video. We're going to concern ourselves more so with just some basic anaerobic pathways that involve something known as fermentation. So again, let me reiterate, cellular respiration produces CO2 and H2O. So as you can see over here, that's what's coming out of respiration and going into photosynthesis. And out of photosynthesis, we get glucose and oxygen going into respiration. But what if there's no oxygen? What if we don't have that there? Well, we're going to have to do something a bit different because, again, we're trying to take glucose, put it through the process of respiration, and produce ATP so that we can provide that ATP to our cells so they can carry out whatever functions they need to do. Everything runs on ATP in your cells. That's the cellular energy currency, so to speak. And cellular respiration produces that. Well, if we have oxygen, we can put it through all the steps, glycolysis, Krebs cycle, electron transport chain, because electron transport requires oxygen. But it gets us lots of ATP, 36 to be exact, for every glucose molecule. Well, sometimes there's going to be a situation where there's not any oxygen, or just not enough. And so it breaks down because those electron carriers, well, now they have nothing to donate their electrons to if we're not doing the electron transport chain. If there's no oxygen, they have to have something to donate it to. And so what we'll see is that all of this is gone. We can't do it. It's not going to happen. And so anaerobic respiration is only going to leave us with 2 ATP. It still begins with glycolysis. So just to recap from our previous video, this occurs in the cytosol, or sometimes referred to as the cytoplasm. It is not in the mitochondria, it's out in the cell, in the cytosol. So in glycolysis, we take glucose. We have to spend some ATP first, okay? We actually have to use some ATP right here first to kick it off, get it started. But then once it runs, we end up producing, we take 4 ADP, and we are able to use the energy that's obtained from the breaking apart of glucose to make 4 ATP. And in the process, we give 2 NAD plus molecules, each a pair of electrons, so that they become NADH. Well, normally, this NADH is going to go to electron transport. But again, that requires oxygen to donate those electrons to. And if we don't have any of that, well, that's not going to occur. And also, some cells, like your red blood cells, for example, well, they don't have mitochondria, right? So what are they going to do when they can't send the pyruvic acid to mitochondria? And what are we going to do when we can't send the NADH to electron transport? The process of glycolysis cannot continue running if you take all the NAD plus molecules and give them electrons and they can't revert back to NAD plus. The process has to have NAD plus to run. So what do? Well, we resort to an anaerobic, no oxygen pathway. And the one that your cells can do is lactic acid fermentation. So glycolysis runs like normal, totally normal. Nothing changes with glycolysis, but the products of glycolysis are going to enter into a different pathway because, again, there is no oxygen. And so these 2 pyruvic acid molecules right here at the end are going to jump over into fermentation And those two pyruvic acid molecules are going to go through a process that converts them into two lactic acid molecules, hence the name lactic acid fermentation. And in order for that to occur, wouldn't you know it, these two NADHs are going to come over, donate their electrons to the pyruvic acid in this process as it becomes lactic acid, and they become NAD plus again, which then cycles back over so that glycolysis can occur very rapidly. So there's fewer steps involved. So this pathway is actually faster than the aerobic pathway, but we only net 2 ATP in the whole thing. That's it. Just these two. That's the only part we're going to get. And so it's no wonder that our muscle cells don't prefer this pathway. It gets them less ATP. It's just not as good. But they can do it in a pinch. If you use up most of the oxygen or about all the oxygen in your muscle cells, exerting them in exercise, they'll revert to this. They'll resort to this and do lactic acid fermentation. Now that lactic acid is actually just going to leave. It'll be carried off in the bloodstream. Okay, so it'll enter your bloodstream, where it will eventually make its way to your liver. And there in the liver, it can actually be reverted back into pyruvic acid, and then the liver cells can just take the pyruvic acid and use it like normal. Send it into the Krebs cycle and let things run like normal. But again, this is occurring where there's not oxygen, or in the case of your red blood cells, they don't have mitochondria. So they can't do the Krebs cycle and electron transport chain. The oxygen they have is also being carried around to the rest of your body. It's tied up in the enzyme the protein hemoglobin, so that it can reach the rest of your body. So they're not using it for aerobic respiration. So they actually do anaerobic. So what about other organisms? Now there are some bacteria that do lactic acid fermentation. In fact, the bacteria that make yogurt, the bacteria that are used in pickling things, and so pickles, sauerkraut, kimchi, yogurt, sour cream, these are all produced with lactic acid fermentation. So those bacteria that can do lactic acid fermentation play a huge role in the food industry. But what about some bacteria or other microorganisms that can't do lactic acid fermentation? What's their pathway for fermentation? Well, that is going to be what's known as alcoholic fermentation. So if lactic acid fermentation produces lactic acid, alcoholic fermentation produces alcohol. So some microorganisms that do this, some bacteria, but also yeast. It's these yeasts, that are used in the process of making alcoholic beverages such as beer and wine. And people have been doing this for thousands of years. They've figured it out, and many have really kind of made it into an art. It's a scientific process, and so the industry, man, they do a lot of different things to kind of adjust and play with and tinker with this process to produce different types of beverages. It's from a scientific standpoint very fascinating. So what's going to happen again is glycolysis is going to occur. We're going to break apart glucose. We're going to get 2 ATP again. So again, this is occurring kind of faster, but we're still only getting 2 ATP, which if you are a microorganism such as yeast or bacteria, that's not as big a deal. You're a lot smaller. Your energy needs are far less, so nobody really cares. They're happy to do this. They'll take that pyruvic acid, and instead of converting it to lactic acid, it'll jump over here and go through a process to become acetaldehyde. I had to think about how to say it again. Acetaldehyde, not a word I say often. And this acetaldehyde, in the process, it's actually going to give off some CO2 gas. That's why Champagne is bubbly. That's why wine vats, when they're fermenting and making wine, they have to have pressure release valves on it, because there's a buildup of gas where these yeast or bacteria or whatever they're using are taking pyruvic acid and turning it to acetaldehyde, and there's CO2 gas given off. And so that's why you get the carbonation, really is what it is. Just like soft drinks are carbonated, that's CO2 gas dissolved into the liquid. Well, there's CO2 gas dissolved in the alcoholic beverages as well. And so once you have these two acetaldehydes, they can then go through a process where this NADH that was produced in glycolysis can jump over, donate its electrons, become NAD+, which can then cycle back over, and that keeps things running nice and smooth so that glycolysis can continue to occur and get those ATP. So the acetaldehyde becomes ethanol or ethyl alcohol, and so those molecules are they're actually C2H6O. And so that is the alcohol. That's the alcohol in the alcoholic beverages. And so again, people just put this to use to make these beverages because they don't require refrigeration really. So for wine, for example, for centuries before refrigeration, you could carry around wine and it would keep. And of course, there's different debates about how high the alcohol content was, because, well, just like humans, alcohol is actually toxic to yeast in certain amounts. And so the type of yeast and the conditions are what are going to determine how much alcohol will be in that beverage, because at a certain point it will kill the yeast as well, and they won't produce anymore. And so let's kind of compare these two anaerobic pathways we've discussed: lactic acid fermentation and alcoholic fermentation, and where. Let's look specifically where some examples of these might occur. Lactic acid, again, I said that occurs in red blood cells. They lack mitochondria, interestingly enough, and so they do lactic acid fermentation. And again, they would carry that lactic acid to the liver, where it would be filtered out and converted back to pyruvic acid. Also in your muscle cells, when you use up the oxygen very quickly. So if you're sprinting, if you're lifting heavy weights, those things that are known as, because they cause your muscles to do lactic acid fermentation and anaerobic pathway, they're known as anaerobic exercises. On the other hand, alcoholic fermentation, this is an example of yeast. They're actually microorganisms in this. And so yeast is an example. Some bacteria do this, and they are used to make beer and wine and things of that nature. This is why if you leave juice sitting out too long, it ferments. There's bacteria that get into it, and it causes it to ferment. It smells horrible and tastes bad. That's why milk curdles. So again, but we've put it to good use, making pickles and things of that nature. Things that produce lactic acid can make yogurt, buttermilk, if you use that for biscuits or pancakes or whatever, those things. So we put this stuff to good use. And just a quick recap to kind of compare the two at the very end. I want to wrap up this video and keep it pretty short. Aerobic is with oxygen. It is, because it has 3 steps, slower, but we get much more ATP. Anaerobic is what this video is about. That's when there's no oxygen used. It only has, this pathway only has 2 steps, glycolysis and then some type of fermentation. So it is quicker, but again, we get far less ATP. And so your cells prefer aerobic, but your muscle cells and red blood cells can do lactic acid fermentation. So I hope that this video has been helpful to you in understanding some anaerobic pathways for respiration to occur, and also comparing that to aerobic respiration. As always, thanks for watching. I encourage you to like, comment, and subscribe. I'll see you next time on Blackboard Biology.